Yes, so our next presenter is Dr. Ying Zhang, and I have followed him for quite a long time because I just found that his way of looking at treatment was just, it's really inspiring. So I want to give you just a little background on Dr. Zhang. He is a professor in the Department of Molecular Microbiology and Immunology. That's with Johns Hopkins Bloomberg School of Public Health. If you're not familiar with where that is on the East Coast, so that's in the Baltimore, Maryland area. His research interests are focused on bacterial persistence, antibiotic resistance, pathogenesis, and development of more effective treatments of persistent bacterial infections, including Lyme. That's a lot of words. But what he's really looking for is what are we doing with those persisters and why can't we break through? And I know that happened in my daughter, it's happened with Logan and hundreds of millions of people who are dealing with this. So I love that he is attacking this. In recent years, Dr. Zhang has been working on developing more effective treatments for persistent Lyme disease and those associated co-infections that we talked about back when we had Nicole looking at all those different diseases that ticks bring. So the group that he's worked with were various FDA approved drugs, as well as some highly potential essential oils and natural products that are more effective than even the current Lyme antibiotic treatments that are out there. For those of you who either can't tolerate antibiotics or don't want to go that way, and you need a more naturopathic approach and essential oils, this is really important. And even for those of you who are those big believers in antibiotics, I think this is an important messaging, that this is another way that you can heal. I was really privileged to be part of Dr. Zhang coming to Harrisburg, Pennsylvania, the capital of our state, when we had a big Lyme rally going on to be able to get insurance companies to approve more than 30 days of an antibiotic. And while he does, look into the fact that antibiotics are still good for those who need them, I love the fact that he talked about empowering us as people and consumers and patients and doctors to be able to look at treatments that are going to be the most effective for their patients. So I'm really thrilled to have Dr. Zhang here. I'm really looking forward to hearing more of what you have to say. So I'm going to turn the presentation over to you.
Well, thank you very much, Carrie, for the kind introduction and for the invitation to present to you. And I also want to really thank Logan for sharing his very inspiring perspective. I'm very much moved. So let me see if I can share my screen. Can you all see this?
Yes, we see it, Dr. Zhang.
Okay, very good. So let me get it into the presentation mode. Can you see it okay?
Perfect.
All right, very good. So what I'm going to do today is to give you an update about our work to develop more effective treatment for persistent Lyme disease. So I'm going to start with the brief overview of the complexity of the disease, and especially the persistence problem surrounding the Lyme disease, which is a huge problem, as we all know. And also the bacteria, Borrelia, that causes the disease can develop persisters, and the limitation of the current Lyme antibiotics in killing such Borrelia persisters. And also tell you a little bit about the work on developing treatment for related co-infection pathogen, Bartonella, as well. So in developing the treatment for persistent Lyme disease, I'm going to use TB treatment as a model because TB is a persistent infection to show the importance of persister drugs. And I will work to identify Borrelia persister drugs and the drug combination approach incorporating persister drugs, as well as essential oils, natural products for more effective treatment of Borrelia bacterial persisters in vitro. This is the in vitro work initially, and then moving on to in vivo in animal testing in the mouse model. Then I'm going to show you this two types of persistent Lyme disease, a new model of PTLDS, where we propose that PTLDS is due to persister inocula, where we divide persistent Lyme disease into two types. There's a type 1 persistent Lyme disease, which is PTLDS. The other is the type 2 persistent Lyme disease, which is untreated chronic Lyme disease, if you like. These were diagnosed late, treated late. Then I'm going to move on to the mouse model to evaluate different drug combinations, these two types of persistent models, so type 1, type 2, as well as reflected in the mouse model as well, showing you some promising drug combinations that are effective not only in vitro, but also in the mouse model. Finally, I'm going to end with strategies for treating persistent Lyme disease and future directions. So as you all know, Lyme disease is actually a complex disease. It actually has different stages, starting with tick bite, early localized form, as you see as EM rash, target-based, although the target is not always target shape. But then it's bacteria can disseminate to get into bloodstream, so the patient can get early disseminated form of the disease, and then moving on to late disseminated form of the disease, such as Lyme arthritis, as well as neurological symptoms. But then there's also this PTLDS, the post-treatment Lyme disease syndrome, which is really the most important problem in Lyme disease, because after all, these are the patient population that are not cured by the current standard treatment. The current treatment is, standard treatment is the IDSA treatment 2 to 4 week, antibiotic treatment with a single antibiotic, monotherapy with doxycycline, or amoxicillin, or cefuroxime. Or if patients don't respond very well in this severe form, neurological form of the disease, then patients can be given ceftriaxone, as well as in the late Lyme arthritis stage. And then the thing is still about 10 to 20% of the patients don't get well. They continue to suffer from fatigue, pain, and neurological symptoms. So these are called PTLDS, post-treatment Lyme disease syndrome. So I have this diagram here, as you can see, although it's not always going this through this stage, especially people now know actually it's largely work of Dr. John Alcott at Johns Hopkins. They did a very careful analysis of these PTLDS patients and show that in fact, quite a significant number of patients actually go straight from the early form of the disease straight to the PTLDS, not necessarily going through these different stages. And then in fact, only about 10% of the patients looks like going from the Lyme arthritis then to the PTLDS, but seems most of them are going straight into the PTLDS. So this is really a lot we still don't understand about this disease syndrome, especially this PTLDS. So there have been different treatment trials for trying to treat this PTLDS condition with a current approach, where we have these 4 published clinical trials done. The 2 actually showed some effect of the antibiotic therapy, 2 didn't. So the one by Klappner et al., where they used IV ceftriaxone for 4 weeks, then followed by 2 months of doxycycline, where they didn't see much of an improvement in symptoms or quality of life. And also the Dutch study, the more recent Dutch study as well, they didn't see much of an improvement. Then there are 2 other studies by Krupp and Fallon, which used 4-week or 10-week of IV ceftriaxone, seeing some improvement in symptoms, but then the improvement is not lasting, so that after treatment for some time, then the symptom would relapse, would come back. So it's not really a stable cure, so it's not really very effective treatment. So there have been a more recent study by DeLong et al. Showed that the PTLDS is a large number of patients suffering from this disease condition, as estimated about 2 million people by this year suffering from PTLDS. So which is a huge challenge facing modern medicine, is we don't really have a treatment. So that's why, you know, you see desperate patients try all sorts of treatment then they didn't quite work very well. And so there's a huge unmet medical need. So what causes PTLDS? So there have been different theories. These four have been the most common ones that are proposed. First one is this host response to antigenic debris. That is, the bacteria already dead, and there's only debris or dead parts of the Borrelia bacteria that get lodged into the joints, into the tissues that continue to stimulate the host immune response. The second theory is this autoimmune disease condition. Then there's not really, infection is no longer there, it's just autoimmune. Third theory is a co-infection that's introduced during the tick bite, or sometimes it could be due to secondary opportunistic infection due to Borrelia causing immune suppression. The last theory is a persistent infection due to Borrelia persister bacteria that are not eradicated by the current Lyme antibiotics. So if you look at the four theories, they're not necessarily excluding from each other. They may actually coexist, but we believe the major driver is still the persistent infection that is not really eradicated effectively by the current Lyme antibiotic treatment. And there is no FDA-approved treatment for the PTLDS condition. So there have been different theories, different studies as well that actually show the persistence problem that shows Borrelia bacteria not killed by the current Lyme antibiotics. There have been animal studies in mice, in dogs and monkeys that have shown this. So I don't have time to go into this except to show you one mouse study that's very interesting in the next slide. But then there are also human studies as well that's really anecdotal. It's not really very consistent. So this is really a very intriguing organism. That is, the bacteria after treatment can remain in a state of viable but non-culturable form, such that when you try to culture, they don't quite grow. But then the bacteria still there can. So this is really the heart. This is really the problem that is facing modern medicine. That is why the other camp, IDSA, the more conservative researchers or scientists do not quite believe this persistence problem. This is largely because the current science is not quite there yet. The current culture methodology is not quite working. It's not able to detect such viable but non-cultural called VB and C form, which is kind of a dormant form. So this is the mouse study done by Amir Hotzik and Steve Bathold, published in 2014. It's a mouse study where they treated mice infected with Borrelia bacteria. So they first allow the infection to establish for 6 weeks, which is sufficient time to establish persistent infection. Then they started treating with ceftriaxone for 30 days. Then they look at the different organs, ear, joints, etc., by different methods of detection, by culture, by PCR. So this is the PCR results. They show that immediately after treatment, as well as after 2 months, 4 months, 8 months, if you look at the tissues, you don't detect anything. They're all negative, as you can see here, occasionally some. But then by the end of 12 months, if you stop the treatment, you can see this actually, the Borrelia bacterial DNA come back. As you can see here, they turn PCR positive. That means the bacteria have replicated, even though you cannot quite culture the organism. So this is really showing the difficulty with this problem. So there's really the VB. So not only in vivo, but in vitro, we and others have shown that in Borrelia bacteria can develop persisters that are not killed effectively by the current Lyme antibiotics. So previously, people's attention are mainly focused on this log phase growing form of the Borrelia bacteria. Can you see my cursor here?
Yes, we see that.
Yeah, okay. So the spirochete form in the log phase, the young culture, then that's actually moving around, but that's not really the most important form of the bacteria. But then when the culture gets older, then it start to develop this aggregated biofilm-like structures. So these are the persistent forms not killed effectively by the current Lyme antibiotics. So here, this published study will show that for log phase bacteria in dark bar, they're sort of killed more effectively by the current Lyme antibiotics. But when they're in stationary phase, not growing, then you can see a lot of the bacteria, higher percentage of the bacteria still remain viable. So here's a slide to introduce to you about the persistent concept. This is not a new concept or idea. It was actually shown back in the 1940s when penicillin was first discovered. It was first demonstrated by this lady called Gladys Hobby from New York. Then she observed that, in fact, penicillin kill majority, 99% of the bacteria. There's always 1%, a small percentage not killed. They called persisters. So this is 2 years later, Joseph Bigger gave the term not killed persistent bacteria. So I use this Ying Zhang model here to demonstrate the two types of bacteria. One is a young, is a growing bacteria. When it grows to a certain stage, it starts to develop the persistent form, the black form. Then when this persistent form accumulates to a certain level, started to develop the growing form, reverters. So this persisters actually show this phenotypic resistance, also called persistence or tolerance. That is, they're not growing, not killed by the antibiotics, by the current antibiotics, but then when you remove the antibiotics, start growing again, remain susceptible to the same antibiotic. So it's not a stable resistance, in contrast to the genetic stable resistance that has a genetic basis due to mutations or antibiotic resistance genes. So this genetic resistance is what we often talk about. Antibiotic resistance has a higher MIC, that is the bacterial growing presence of the antibiotic. But in persistence, the bacteria don't grow. I call yin resistance. They don't grow in presence of the antibiotic, but they're not dead. When you remove the antibiotic, it can cause relapse, can grow back. That's why clinically, in fact, we have a range of persistent infections that are due to this condition. For example, urinary tract infection, TB, tuberculosis, biofilm infections are typical examples of persistent infections, and they can have post-treatment relapse. So I'm going to show you the TB treatment as a model for more effective how to develop a better treatment for persistent Lyme disease. So then also people have identified the persisters, as well as there are different, even the amount of persisters, they're not all the same. They are very heterogeneous. They have different forms of persisters. It's a spirochete form of the persistor, round body form of the persistor, microcolony form of the persistor, biofilm form of the persistor. So these are very heterogeneous. So this important concept of this heterogeneity of persistence, they can develop different levels of persistence, not only in vitro, but in vivo as well. So I'm going to move on to this TB treatment. So currently TB people treat TB using this drug combination approach of 6 months with 2 phases. The first phase, intensive phase, was 4 drugs: azanazid, rifampin, pyrazinamide, which are highlighted in red, yellow, because it's a persistent drug, important. Then ethambutol. And then have a second phase, continuation phase of azanazid and rifampin, as this is demonstrated here in the Ying Zhang model. So the TB people know that you have to use a drug combination. Using different drugs kill different bacterial populations. Like azanazid, the drug kills only the growing bacteria. Rifampin is a drug kills both growing and non-growing bacteria. Then we have pyrazinamide, PZA, that kill both. They actually kill only the persistent bacteria. So TB people over the years, 70 years, long time, developed really a lot of experience treating this persistent infection. It's a drug combination that people learned 3 lessons. So the first lesson is that the drug combination is not only to prevent the emergence of resistance, but also to enhance the efficacy of the treatment. So in the 50s, people know that if you use single drug like isoniazid, get a lower percentage of cure, like a 3-month sputum conversion, only 37%, in contrast to 67% if you use a drug combination of streptomycin plus isoniazid. So we know the effect drug combination, okay, to enhance the efficacy as well. So the second lesson TB people learned is that you need sufficiently long in order to cure the persistent infection. This is very important. So back in the 50s, the treatment of TB with the 3-drug combination, streptomycin, PES, and isoniazid, if you treat for 6 months, you get 62% relapse, very high percentage of relapse. You treat for a year even, you get a close to 20% relapse. You treat for 2 years, get a low percentage of 4 or 5% of relapse, which is acceptable. That is why in the 50s, people treat TB for about 2 years, which is a very long time. So, but it's really the introduction of persistent drugs in the treatment of TB field, initially with the rifampin. Okay, adding rifampin as a minor persistent drug, shorten the therapy from 24 months to 12 months. Then it's adding pyrazinamide. It's the very important persistent drug. Then shorten the therapy by another half to 6 months. So then TB people know the importance of persistent drugs. So that's lesson number 3. So in TB, then we have a persistent drug, pyrazinamide. Another important persistent drug is called clofazimine, which is a leprosy drug now used for treating MDR TB, as well as its actually potential to shorten the TB therapy that people. These 2 drugs happen to have activity against Borrelia persisters. So I'm going to come back to show you the combination with current Lyme antibiotics with these 2 TB persistent drugs actually turn out to be effective for treating persistent Lyme disease as well. So this is really a cartoon. Some of you may have seen this really to demonstrate the importance of persistent drugs. And so this is the current antibiotics, like a mower chop off the top part, which I call dandelion phenomenon. You still have the root here, which is a persistent bacteria. So when you remove the antibiotic, you stop the treatment, then because the root is still there, you get relapse. You can get relapse. But of course, sometimes you do not. This is very heterogeneous. So, but then what you need is a shovel that dig out the root, kill the persistent bacteria, then the whole thing dies. So the pyrazinamide is one such drug like the shovel. So in actually to treat more effectively treat persistent infection, you need the combination of mower and the shovel. You need a combination approach. So that's why in treating persistent Lyme disease, we propose to apply the PZE principle for treating persistent Lyme disease along with other persistent infections. So this is shown here in this Ying Zhang model. And so when we started try to address the problem, we don't have any drugs. We didn't have any drugs active against Borrelia persisters. So we have to develop a rapid viability staining called a cybergreen PI assay. And then we screen different drug libraries, FDA approved drug library, etc., where we identified a range of drug candidates. These were all published. So I'm not going to go into details except to tell you that we identified some that look quite interesting, have a much better activity than the current Lyme antibiotics killing Borrelia persisters. So this is actually to show you the cybergreen PI viability assay we developed. So this is shown here in panel B, that where the live bacteria stained green, dead bacteria stained red. So when you apply an antibiotic, if the more cells turn from green to red, that's a good sign. The drug is effective because killing the green cells, live cells, then turn into red dead cells. So using this assay, we're able to rank the relative activity of the current Lyme antibiotics, as well as in comparison with our persistent drugs we identified. So several interesting points I want to show here. One is that tetracycline is more effective than doxycycline. So the lower the percentage, the more active the drug is here. We're looking at the percentage of viable bacteria. So here, that's actually correlates with clinical observation. Studies by Dr. Sam Donta actually showed some interesting study that tetracycline is more active than doxycycline clinically. There, as well as we know that ceftriaxone is the most important, most powerful current Lyme antibiotic against Lyme disease. In fact, it's actually the lowest percentage. The others, cefuroxime also showed pretty good activity here as well. So here we use the same micromolar drug concentration, so we're able to rank them, rank their relative activity. Also, among the macrolide antibiotic, the clarithromycin is more active than azithromycin. And importantly, our persistent drugs, in fact, the daptomycin, clofazimine, which I mentioned, this drug is a leprosy drug, MDR-TB drug as well, turn out to be very good, actually much more active than these existing Lyme antibiotics, and also some sulfa drug as well. So if you look at these persistent drugs, they don't work the same way as the current antibiotics, because they actually preferentially act on the membrane. Cell membrane is very important for persistent survival, as well as for example, this daptomycin, clofazimine, essential oils can all potentially act on the membrane of the Borrelia bacteria that can kill the persistent. Membrane is very important for persistent bacterial survival. Without the membrane, they die. So the other important feature of the persistent drug is that they damage DNA or produce reactive oxygen species that can damage DNA. For example, this anthracycline antibiotic, anti-cancer antibiotic, very powerful. Donomycin can damage DNA, kill Borrelia persistens very powerfully. Clofazimine has some of that effect. Cephalosporin have some activity there as well. So then there are other agents like sulfa drugs, quinolone, rifabutin. Actually, those can block DNA RNA synthesis. They had some activity as well, along with some macrolide antibiotic has some activity also. So moving on to this further evaluation of these drug candidates, in fact, this is a study done by a master's student from my lab, Hector, is doing a great job as part of his SCM thesis, where he evaluated some of the drug candidates, especially, I know there's a lot of interest, Dr. Rajita is going to talk about his findings about disulfiram and azlocilline. So Hector actually compared these. This is actually all these drugs is a stationary phase bacteria, old culture treated with 50 micromolar of these drugs, then for 1 week, then look at the survival rate. And as you can see here, these are the current Lyme antibiotics, and then we see actually the azlocilline and disulfiram have similar activity, and they don't really have a very strong anti-persistent activity, certainly not as strong as the ones we evaluate, like the daptomycin, clofazimine, even clarithromycin. The other one we found quite active is cryptolipine. It's the active component of cryptolepis, which is a natural product, which I'll come back to. So if you look at the activity of these drugs against the growing form in terms of the MIC, against the growing form, then these two drugs have good activity there. For example, azlocilline is more active than amoxicillin, but is not really as active as ceftriaxone or even clarithromycin. Disulfiram has activity against the growing form. Yeah, it's 0.3 to 0.6 microgram per ml. Yeah. So according to our study, it looks like these two drugs have good activity against growing form Borrelia, but not so good. Had some activity, I would say, not so good as against the persistent forms. So this is actually the microscopy data that corresponds to these percentages, where you can see the green, the control untreated, a lot of them are live, 92% live cells. Doxycycline, you get a lower percentage, 67% live cells. But then you have some other daptomycin is very good, 0.2% viable cells. Clarithromycin, dalsulfam is the data I showed you. You still have some green cells remaining that has some activity, better activity than doxycycline looks like, but not really as good as some of the others we evaluated. So we found that even though we have the persistent drug, we can't use them alone. They have to be used in the context of drug combination, as I showed you in the Yin Yang model. So we need, or in fact, we found the most effective one is a 3-drug combination. So once we found it's a daptomycin, doxycycline, cefuroxime. But this was all published, so I'm not going to go into detail except to say that even though this is powerful, daptomycin is a very expensive IV antibiotic. We're interested in develop to replace daptomycin with oral persistent drugs that are equally or perhaps more effective than this combination to kill the persistent forms. So this is actually just some additional slide to show you the combination of some of the drugs we evaluated that show promise. You can see red, that's what you like to see, because the red indicates the bacteria are dead. This is our control, so this is actually when you combine the 2 current Lyme antibiotics together, this is untreated, the treated, that is a 40% still viable, but this is our triple drug combination, the daptomycin drug combination we use as a control. And these are the 3 examples we showed that have good activity in combination. Is a clarithromycin plus nitroxyline, as well as linezolid, and this is a 3-drug combination. So this is the slide we published earlier where we also evaluated different drug combinations, including HCQ. You all probably know about this, is a hydroxychloroquine, which is really the drug used for treating COVID-19, has shown some promise there. And it's interesting, it's also used for treating Lyme arthritis. So some of the Lyme patients may take HCQ, hydroxychloroquine, that may actually protect them against the COVID-19. So I thought it would be of interest in such a current environment to mention this potential interesting observation. So then we went on to look at some other treatment possibilities, looking at the natural products like essential oils. So we've done 2 studies on essential oils. First one, we identified 3 most active ones like oregano oil, cinnamon bark, clove bud have a very good activity. Then in the second study, we identified another 10 of them look very good. The garlic oil, allspice, myrrh, Hadicam, Lycicubaba, etc. And we showed that, in fact, these essential oils in combination with the current Lyme antibiotics has shown good activity, like all these. So I'm not going to go into detail. So this is, by the way, I want to just tell you that this is all in vitro study so far, okay? And I'm going to tell you about the animal study later. So also evaluated this natural product and the herbal medicines. These was done in collaboration with Dr. Sanjay Schweig from California, as well as Leon Jacob. Jacob Leon. So where we evaluated a range of herbal medicines that are used by Lyme patients. This is largely derived from the Buhner book, this Lyme herbal medicine book, where we evaluate a panel of about 14 natural products. It's interesting. And we found actually 5 of them to have very good activity against Borrelia persistent forms, as well as against the growing form. So the one, for example, cryptolepis, okay, is very potent, very active. The other one is the Japanese knotweed, which is an invasive species in the UK. They try to get rid of it. So it's a very good plant that has good anti-Borrelia activity. Then the other one that's pretty potent active is black walnut. Cat's claw as well is very good. Artemisia annua, the herb, is also has some activity, but not as strong as the ones I highlighted. So the ones I highlighted in yellow turn out to have activity against Bartonella as well. So this is a study which not yet published. So this is the study, and now that I'm talking about Bartonella, we also did FDA drug library screen against Bartonella because it's an important co-infection pathogen. So these are actually currently used antibiotics for treating Bartonella infections are listed in black here. The aminoglycoside, this macrolide antibiotic, rifampin, doxycycline, etc. Then we found those highlighted in red to be more potent, more active than the current existing Bartonella antibiotics. These include daptomycin again. So we get this one drug that has activity against both Borrelia persister and the Bartonella persisters. Now the interesting one is methylene blue. It's actually very potent, very active against Bartonella persisters. The azole, antifungal azole drugs, nitroxyline, the antibiotic for treating UTI, has some good activity as well. So in drug exposure studies, you can see those are the really kill completely the bacteria in terms of colony count. So what we do is that we expose an old stationary phase Bartonella culture with all these different drugs at 50 micromolar. Then at different time points, 1 day, 3 day, look at the number of bacteria that survive. So you can see those highlighted in pink are the ones that are most potent: methylene blue, daptomycin, azole drug, nitroxyline. So among the existing antibiotics for treating Bartonella infection, the ones aminoglycoside, amikacin, kanamycin, pretty good. By day 3, kill all the bacteria. And the other one that's active, second to aminoglycoside, is rifampin. Okay, 10 to the 3. So all the others are pretty much similar. So this is all in vitro. Then we also went on to evaluate the essential oil panel on Bartonella, where we found again some of the overlapping essential oils have activity against Bartonella stationary phase form as well, like oregano, cinnamon bark, clove bud, allspice. All this highlighted in red are the ones. Now I'm going to move on to the animal studies, where we have 2 types of animal models. One is this long-term called type 2 persistence, which is long-term persistence model, which is a study established, the model established by Amir Hotzik and Steve Bathold at UC Davis. This is a study we're doing, evaluating different drug combinations with our collaborator, Monica Embers at Tulane. So got some data there. So I'm going to talk very briefly about this because this is not published. Then our published work on this, using this type 1 persistence model, starting with a persister inocula, then develop a more rapid persistence model, which is a Lyme arthritis model. So with different inocula, so this is where it's important. I think previously people's attention focused on this log phase, spirochete form of the bacteria, but we believe that may not be the most important form. And then in the older culture, they developed this round body form as well as the microcolony aggregated form. These are more tolerant to persistence. So the current Lyme antibiotics don't kill them very easily. And these, not only that, not only tolerant to antibiotics, they also cause more severe disease. So it's actually shown in the mouse model here. This was published earlier last year, March 2019, where we evaluate these different forms, the log phase, stationary phase, as well as microcolony form, injected into mice. Then at day 21, you can see the microcolony form causes more severe arthritis, swollen, more swelling in the joint. But the log phase is not yet able to cause very severe arthritis on day 21. But on day 35, the difference between these two groups getting smaller because this log phase, stationary phase that caught up, they actually develop a more severe arthritis, but still the microcolony-induced arthritis still remain more severe, although even though it can go down in severity as time goes on. So this is actually also shown by histopathology, heart base, as well as joints. So I'm not going into detail here. I'm going to move on. So based on this mouse study, we propose that in fact there's two types of persistent Lyme disease alluded to earlier in my presentation. So this is a type 1 persistence, this is an early development persistent Lyme disease. This is essentially PTLDS. That is, this is a type of persistent infection that's introduced very early during the tick bite. So depending on the Borrelia bacteria in the ticks, so the hypothesis is that the Borrelia bacteria residing in the ticks are very heterogeneous. So it's depending on the particular Lyme bacteria being introduced into the body that can determine the disease outcome to some extent. So if it's a log phase growing form of the bacteria, that can be easily killed by the current Lyme antibiotics. So the patients are cured. So that's why you get the 80 to 90% of the patient population cured the current acute form of the disease. But in some cases, people may get unlucky because the tick may carry this biofilm or persister form of the bacteria that's introduced. Even though you treat with a single antibiotic, you don't cure it. We know biofilm infection cannot be cured with a single antibiotic. So then you have the late development persistent Lyme disease. That is, the initial infection was not diagnosed early enough, treated on time, then developing into this persistent form of the disease. So using this type 1 persistence mouse model, we evaluated a different mouse model. We developed this log phase, the spargate form introduced infection, as well as this microcolony, the persister form introduced infection. Then evaluated different drug combinations. Then we looked at culture as well as PCR of the tissues. So by culture, we can see the microcolony introduced form produced a more persistent infection that's not easily eradicated. Of course, majority of them are. You can see there's a number of mice that are positive still remaining. So microcolony introduced foreign infection, ceftriaxone treatment still has one mouse remain positive by culture, but then by log phase form introduced infection that's easily cured. Then we show that in fact the daptomycin drug combination is able to eradicate the infection by culture as well as by PCR. So the PCR data is shown in brackets in here. So this actually validates our in vitro finding in the mouse model. So here I want to really mention this very important concept of these different levels of persistence. So you can produce a mouse model that is easily cured with a current Lyme antibiotic, as well as you can establish a more persistent infection that the current treatment, like even ceftriaxone, the most powerful antibiotic, cannot eradicate. So it all depends on the model. So now moving towards the end, in fact, so for the future treatment of Lyme disease, so in the short term, I think we can use this, still use this IDSA treatment, but then we'll have 10 to 20% patients not cured. So these are the candidates for testing the persister drug regimens we identified. But in the long run, once we identify some of the optimal persister drug combination, they can potentially use to start from the very beginning so that we don't even get the 10 to 20% patient not cured. So we can get a much better cure for the disease. So to move forward then, so I think even though we identify some promising drug combinations, I want to emphasize this is a long-term thing, and we still need to evaluate all these different drug combinations, especially the combination essential oils, because there's a potential side effects of the chemical drugs, then some natural products as well as essential oils look promising. these need to be examined as well in terms of their active components, their utility in vivo in a mouse model. Now that we identify some promising drug candidates, I think it's important to move on to clinical trials to evaluate these persistent drug combinations. And it's important to determine, try these on appropriate patient population, as well as we have to know really how long to treat. We can't just treat for 30 days because the 30-day is what we treat for active disease. For the persistent form of the disease, you need to treat longer. But the optimal length of the treatment, I think we need to determine clinically by the proper designed clinical trials. And then we need to identify biomarkers for treatment response, as well as address the co-infection problem and the immune suppression. And we can't do all this without funding. And finally, I really want to end my presentation with this Ying Zhang model, which is really try to bring some unity to this field. That is to propose this Ying Zhang model to try to unify the field, where Yang means the active form of the disease. That's the current treatment helps. But then we have the Yin, the persistent form of the disease. Then we need a different approach, which is a persistent drug combination as shown here. So this Ying Yang model actually also shows, not only sort of tells you about the two forms of the disease, this active form of the disease that's really taken care of by the IDSA approach. Then there's also the persistent form of the disease that's really largely pushed to this ILADS physicians, LLMDs, who try to take care of these PDLDS patients. But then this model also provides a solution to this problem. That is, we need a persistent drug combination, which we found some, we think have potential to really bridge the gap and to really solve the problem facing the field. Try to solve that. I mean, as we all know, this is really a war. It's really partly because the current science is not quite there yet. We don't quite know the persistent infection very well. We're only focused on this more acute form of the infection. This is not only unique to Lyme, in fact, many other persistent infections. In fact, this is where it leads to chronic form of the disease of unknown etiology, autoimmune diseases, diseases of unknown etiology, like CFS, chronic fatigue syndrome, MS, Parkinson's, you name it. So I think, I often get asked the question, where do I stand in this debate? I can tell you, I stand on both sides. I stay here, right here in the green dot. So I think with this Ying Yang model, we'll be able to unify. I think the more important thing is that if we are able to really show the efficacy of the persistent drug regimen, then that would actually help to resolve the controversy and help patients, I think, above all. So finally, I want to thank the people who have done the work from my lab, GFN, and many others who have done excellent work. And also my collaborators, Monica Ambers, Mir Hotzik, and also the funding, different funding sources. So I'm going to stop here and be happy to answer any questions. Thank you.
Thank you so much, Dr. Zhang. I have heard you give this talk, and each time more is uncovered to me, and it's just I find that I love your Yin Yang model. I think that's really where true healing is going to take place. So we have several questions that have come in, so we'll take a few minutes to answer those before moving on to our next talk. One is the impact on the body different from European Lyme, and are they treated the same?
European Lyme have some unique features. They can tend to cause more neurological symptoms, like the Guerinii form of the bacteria, and then Afzelii cause mostly skin chronic form of the disease. But mostly, I would think the treatment would apply to all these Borrelia sensualatus species. I call it the Borrelia burgdorferi sensualatus species. That is Borrelia burgdorferi, Borrelia afzelii, guarinii, etc. Such approach would be effective against the other forms as well. Yes.
Great. Thank you so much. Getting another question. I'm hoping I'm going to pronounce this correctly. Ivermectin was recommended treatment for my son after discovering large amounts of biofilm in his blood. Have you any information regarding that?
Yeah, we haven't really evaluated ivermectin per se, but I believe it's in our FDA drug library. We screened, but it didn't come up as a very active candidate. So, but that can be examined, that can be looked at.
Great. Another one. I'm wondering about the effectiveness and rationale for pulsing antibiotics such as Dapto.
Yeah, so we done some in vitro work where we compared pulsing of this. So the pulse dosing, then it looks like it works only for cidal drug, like cephalosporin type of antibiotic, but not so much for this static bacteriostatic drug like doxycycline. So we showed that it's really very complex. So that's why I didn't really have time to go into the treatment, the complexity of the treatment. So it's a very complex issue there. You can't really rely on a single drug, a single antibiotic to cure a persistent, very complex persistent infection like Lyme disease. So that's why, you know, I think this pulsing approach has merit, but then also it all depends on how you do it. And by pulsing, then there's a pulsing of the same drug or it's a different drug and all that. So rotating different drugs, this is all sort of a open question. I think this is may potentially help, but I don't know unless, you know, more conclusive studies are done. We don't quite know that. But I think I know some physicians have some experience with that, but this really needs to be validated.
Thank you. All right, we have a few more coming through chat as well. One comment, may we get this lecture into all the medical schools or continuing education for physicians and other ancillary healthcare providers. So we think that this is critical information to be shared. Another question is about patients who've been diagnosed with rheumatoid arthritis and have been treating the arthritis unsuccessfully. Could it be possibly that this is untreated Lyme, and should they be then following the protocol that you recommended to kill persister cells in order for that RA to be effectively treated?
Well, this is a very important question, in fact. So in fact, the study done by Dr. Alan Steer's lab recently, I think it was a year or two ago, they published a paper where they followed Lyme arthritis patients. In fact, some of them develop into rheumatoid arthritis that fulfill the diagnostic criteria of rheumatoid arthritis. So that means at least a portion of the rheumatoid arthritis could derive from Lyme disease, Lyme infection, Lyme arthritis. Then after some time, then when the infection is not resolved, it can develop in autoantibodies that can fulfill the diagnostic criteria for rheumatoid arthritis. So in terms of the treatment, that's what I think. This is also only at the experimental stage. I can't quite, I'm not a physician, I don't really recommend anything. So this is just based on our research. But I think it's really up to the particular patient and the physician to utilize any of the information. And this is like the COVID-19 situation. Mm-hmm. There's a high debate about hydroxychloroquine. On the one hand, you have this, you know, group that says that you should use hydroxychloroquine even without FDA approval. Of course, we now know this FDA approved the emergency use. But then there's the other camp that says, no, you can't use this because you haven't done proper clinical trials. But we all know the trials take years, right, to complete. So that's why in the meantime, patient can't wait. So I perfectly understand, although it's not really up to us to say whether they should try it, but it's really up to them. So I perfectly understand if someone wanted to try it, but
great. And I know this compassionate care that they're bringing up with COVID-19, and you hear, the Lyme community saying we've been begging for these things and for doctors to not be in trouble when they do try those things. And so your research is critical for that. So we are blowing up with lots of questions. I'm going to take about 5 more minutes to do that. What we will do is, if we can't get to your question live right now, what we will do is I will pass along these questions to Dr. Zhang, and then we will send a follow-up email with answers to questions we couldn't get to, if that sounds good to everyone. So getting lots of questions about different drugs and different pieces. One question for clarification. They're trying to understand, did you say that doxy is not effective? And what my understanding was is that alone doxy is not. But if you could speak to that.
Well, doxycycline is a bacteriostatic agent, but then under some conditions can show some side effect, but it's not really very active against the persister form. But has good activity against the growing form, and then in vitro, but then for the stationary phase form, it's not very active. And this is also in vivo in the mouse model as well, but depending on what kind of model you use. And but anyway, yeah.
So I'm also getting some questions about the use of mitomycin C and daunorubicin for persisters and log phase. Would these work for Burgdorferi and for myomytoi? I am so sorry about the butchering of some of the medical terms.
No, I think, yeah. So mitomycin C in our evaluation is not really as active as anthracycline, daunomycin, doxorubicin. And it had pretty good activity on against Borrelia persister form. But then because it has a potential side effect, so that's why it's not really often used. But then the activity, we haven't really evaluated their activity against myomycin, but we would expect it would have the same activity. Yeah.
Great. And just a couple more in our time frame. We got 2 more minutes before we move on. And what about using anti-cancer drugs, repurposing them for bacterial infections such as Lyme and the co-infections?
Yeah. So as I said, so unless it's under very extreme conditions when current things don't work and the patient or may suffer some comorbidity condition like cancer, if they wanted to try it, I think, of course, I'm not really giving advice, but I'm just telling you based on our work, daunomycin is really amazing, very powerful, very potent cancer drug. But, I don't know. So it's really up to the physician and also the patient.
And we do have a lovely friend of ours, Dr. Neil Spector, who was not able to join us. I know he's doing quite a bit of work in that, looking at his work in the cancer fields and treatment of Bartonella. So hopefully
Right.
TikTok 2021, we'll have Neil in that can talk more to that. And we'll give just one more question here, I think that could be helpful. We'll get 2 more in. Looking at MS and Lyme and so they're just talking about persisters and nematodes and in the brain, such as in Lyme and brain issues and things. It's really bad in Australia. So do you have any thoughts on the nematodes and in the brain?
Well, I don't know. We haven't really done work on nematode, but I guess we don't know that, and that can be evaluated or should be evaluated.
And final question, if you don't know if your tick bite results in immediate introduction to persisters or biofilms, why not treat that immediately with medicines for persisters? Is there a harm in doing so?
Yeah, so that's what I mentioned in my slides towards the end for the future. So initially, if we can really identify good persister regimen that really works well without much side effects, that can be used from the very beginning, so that we don't even get the currently 10 to 20% patient not cured. by a single drug like doxycycline, so that we can eliminate that 10 to 20% or reduce that significantly to say less than 5%. So that's possible with a persistent drug regimen, yes.
Okay, great. And this is more of a comment that is from someone from Joy, and I think is something we should all say. We want to pass on our huge thanks to all the presenters for their continued work to fight this horrible, devastating illness. So thank you, Dr. Zhang, for all the work that you're doing. And every time I hear you, I learn more. So I so appreciate all that you're doing for the community and for your willingness to continue to come and share what you're learning.
Thank you. Thank you.
Thank you.